Okay, hello everyone. Hope everyone is doing great. I wanted to jump in here and cover few material that is important for your second midterm. So your second midterm is on a week from this Monday. And then as you know, it covers a lot more material than your previous test covered. So if I were you, I would start studying as of now. So I'm going to cover a few material, but before that I wanted to mention something else. Sometimes we don't get to respond back to your emails because simply because there's 500 of you and there's only 2 of us. So if you haven't heard from us, or if you send us an email and we haven't responded back, please, after 1 or 2 days, please resend your email again. Hopefully this time we would get back to you. So, but what I'm saying is it's easy to get, to skip emails because we receive a bunch of emails. So if you haven't heard from us, please do send it one more time. So with that being said, I wanted to cover a few topics today that are really essentials and are really important for your next midterm. I'm going to cover action potentials. I'm going to cover summation, temporal and spatial summations, and agonist and antagonist. And these are really, when I was learning it a few years ago, when I was back in, as an undergrad, it really fascinates me because I really could relate it to, okay, this is how the brain works. This is how the neuron works. And it always fascinated me. And I always love to talk about it. So let me go ahead and share my screen here with you. Let's see. Share screen. And I will be using a whiteboard. Okay, so hopefully you can see this and everything is okay. Let me just make a few changes. I always love to write on if I can figure it out, let's see, on a blackboard, because it makes the contrast makes everything more visible. Okay, here we go. So today, the first thing that I want to talk about and I want to cover is the neurons and action potentials. So let's talk about, so the first topic is going to be neurons and action potentials. It's kind of a weird way of teaching because it's a lot time consuming. I have to type and I have to draw. Okay, but let's do this. Okay, so neurons, change the color here. Let's do yellow. Okay, let's say this is the neurons. This is the soma, and excuse my drawing. And this is the nucleus, right? And we have a little branches coming off of our cell, which they are called dendrites. So what they do is they receive the information from another cell or another neuron terminals, and they transfer the information that they receive into the cell, and it causes the action potential. And we are going to talk about about this in detail. And then here we have our axon. Okay. So again, this is soma. This is the nucleus, and these are the dendrites that they receive the information from another neurons. And then we have an axons here, and axons are covered with myelin sheets. Let me do pink. And so these myelin sheets, it's actually really difficult. Okay, and we have myelin sheets. What this myelin sheets does in an axon, it helps the axon transfer the information, the electrical potential, the sodium. It helps to transfer the information much faster. Actually, there are a lot of disorders and difficulties comes when the myelin sheets are damaged or they're not working properly. And at the end of our axons, we have what we call, that was not good. Let's see, how did I draw it? Yeah, we have axon terminals, which the information that has been sent to the cell, the electrical potential that has been sent to the cell and transferred down the axons is transferred to another neurons, right? So these are axons terminals, right? And just to be absolutely clear, we might have, these might be connected to another cell body or another soma that has dendrites coming off of it, right? So the neurotransmitters are released and are binded to the receptors on this dendrite. That's how the information is getting transferred from cell to cell. And it's really a fascinating process. Once you know more about it, you always want to know more. How does this work? How does that work? Okay. So, so again, we have our dendrites. Their job is to receive the information and transfer it to the soma, to the cell. And we have axons, which the electrical signals that has been received by dendrites, when they are positive enough to make discharge in the cell, they are getting transferred down to the dendrites, and it causes the dendrites, the neurotransmitters in dendrites, to release and get accepted by dendrites of another cell. Hopefully that didn't confuse you too much. And then on the axons, we have myelin sheets. And so now let me explain how the whole process of action potential works in axons. So we have the axons, and then we have it's covered. The axon is covered by myelin sheets. So let me go ahead and draw axons only here. So I'm going to attempt to draw the axons only. So what happens in the axons, we have the nodes here that we don't have, the myelin sheets are not covered. The little spaces, we called it a node of Ranvier, right? And what simply these nodes of Ranvier is, these are ion channels that allows the inner cell communicate with external cell. So the external cell, what we call which is outside of the cell, it's positively charged. So we have a bunch of sodium here, Na. We have a bunch of positive sodium here that are floating around, and inside the cell we have a potassium, which also have a positive charge. But the thing which is important is that the outer cells, the outer environment, has a more positive charge than the inside one. So the inside one has a more negative, you know, voltage. And this is what the resting. This is the resting point. This is the you know. The resting point for the cell. They are comfortable this way. Now, because of this voltage difference, so the voltage that we have here, it's around, let's see, around negative 70 millivolts. And so this is the resting place for the neurons. So now, what happened when the dendrites of the cell receives some information and is passed to the axons? It caused a displacement of the ion charges. So what that means is that we have a bunch of ions, we have a bunch of channels that are sodium potassium sensitive. So once the displacement happen within the axons, when the electrical charge is going down, the action potential is created by this ion channels opening up and allowing the Na, the sodium, the positive charge of Na go inside the cell, right? And then we also have let me do that in different color, and we also have potassium channel that, because now we have a more positive charge inside the cell, causes the potassium it kicked out a potassium into outer cells in order to maintain the positive negative balance of of the environment. So this process causes a action potential. So if we have time, and if we have piece of membrane potential, right? So when this happens, when electrical discharge happens, what I mean by that is that when the cell receives some information from another cell, terminals of axons of another cell, when they receive some information, it creates a little discharge, and this discharge, as this happens back to back, adds up, and it creates a action potential. And then within an action potential, we have the depolarization and we have repolarization. This is when the potassium is kicking out. This is when the sodium goes in, and we have a more positive positive charge inside than outside. This is the depolarization. And now the process of getting back to that resting point, to what cell does, what axon does, there's ion channels, it kicks out some positive charges, which are potassium, to reach that stable point. So it does that so much that the cell becomes more negative. So it goes to the hyper polarization, which the cell, it's more negative than negative 70, but it reaches back using these ion channels to get back to the resting area. So this is the process of action potential. With that being said, and you might ask, why is it that the cell, we have more positive charge outside than we have than inside? Because these channels are sodium, they're more permeable to sodium. So they let 3 sodium in. Actually, sorry, they let 3 sodium out, and then they let 2 potassium in when they're trying to reach the stable resting point. Okay. So with that being said, so again, what is action potentials? Action potentials is the neuron's way of transporting electrical signals to each other. It's a way of that they are communicating with each other, and it's a fascinating process. So let's see. Um, so that's that. So that's the action potentials. I mentioned, let me just check to see if I mentioned everything that I wanted to mention. Yes. Okay, now let me talk about a synapses on synapses structure. So once the action potential releases, it causes the little neurotransmitters that are inside these axon terminals to get released. And how that process works. I'm gonna zoom into this area right now, actually right here from, I'm gonna um, focus more on that part so I can explain. Let's see. Okay, page. Let's do that. Let's do, okay. So now we have axon terminals. And I'm just gonna draw, wow. I'm just gonna draw one so it doesn't confuse you too much. So this is one axon terminals that is connected to the main axons. And then we have I don't know, we have another uh, dendrites of another cell that they all have receptors, specific receptors. Okay, and so this area, the little, they're not connected, they're not touching. There's a little area in here which is called I have to type that, which is called synaptic cleft. Synaptic. Okay, so this area is a synaptic cleft. It's an area where the neurotransmitters from this axon terminals are released into the synaptic cleft and is being picked up by these receptors. Okay, and let me, you know what? Let me actually use. Can I delete it without making a mess? Yep. No. Okay. Getting used to this. One more. Okay, perfect. I'm gonna use a cell. I'm gonna draw the cell like this because that's, that can I can explain that better. Okay, and this is the cell, right? And you can assume we have a dendrites. So now inside these little axons, right here, axon terminals, we have vesicles, right? We have vesicles that they hold what we call neurotransmitters. I can put little dots, but you get the idea. So these vesicles, they hold neurotransmitters. So what happened is that as this action potential travels down the axons, and when we have these ion channels letting sodium in and potassium out, and we end up with a slightly more positive charge than outside, what happens is that we activate another channels I'm going to do that in blue, which are calcium. We also have calcium outside our cell, uh, which has a positive charge, right? We also allow that to go inside our cell, making it even more positive. And you might ask, why is that? Because, well, isn't it that the cell is trying to reach a stable point, a resting point? And the answer is yes, but the calcium is needed in order to release these neurotransmitters. And we have a special protein. We have a special protein that works as those of you who are bio major knows this better. We have ATP, and this ATP helps to flush out the excess calcium, right? So now the calcium goes in and binds to, let me change the color to the blue, binds to the side of the vesicles that holds neurotransmitters. And these calcium causes these neurotransmitter to get released into the synaptic cleft, right? So now, let's see, I have to keep changing the color. Okay, so now let's imagine that these are the neurotransmitters that are getting released into the synaptic cleft. And this is the main idea. This is exactly what happens when we were talking about anxiety, when we were talking about depression, when we were talking about stimulants. This is exactly, it has to do with neurotransmitters. Some drugs are speed up the process of releasing the neurotransmitters. Some drugs are like SSRIs, like Prozac, and most antidepressant drugs that are out there are SSRIs. That means there are serotonin, which is a form of neurotransmitters, reuptake inhibitor. That means it doesn't allow the neurotransmitter to get absorbed back, which as a result we have a amount of neurotransmitter serotonin or dopamine or norepinephrine available in the synapses. So it decreases the feeling of fatigue or depressed mood and so on and so forth. And so these neurotransmitters will get attached to this cell or to the neurons, and causes the ion channel that are here to open up. So we have sodium ion channel, like the same thing that I've mentioned before. We have sodium ion channel, we have potassium ion channel, and these are open up, and it helps to communicate with the synaptic, with synaptic cleft that we hear. So once the neuron is attached to the cell, it causes the sodium channel to open up, and the sodium channel goes in. The inside of the cell is going to be positively charged. It triggers the action potential down the cell, and so on and so forth. And another action potential, and this is how we feel the way we feel, how we perceive, how we hear, and this is actually the concept of it. So hopefully I didn't confuse it too much, but that was the neurons and neurotransmitters, and so hopefully you understood that. One thing that I wanted to mention. So now this cell, this axon, the terminals right here, it's called a presynaptic membrane. All right, we have this is a presynaptic membrane, because if we call this synapse, then what the neurotransmitters are within we call it a presynaptic membrane. And once the neurotransmitter are released into the synaptic cleft and attached to the membrane of the cell, this membrane of the cell is a post-synaptic membrane. So these are the terminology. And let's see what else I wanted to cover. It's fun to know that the space right here, this is not at your level, but it's fun to know that this is a really tiny space between the cell and the axon terminals. This is about 20 nanometers, and this is extremely small. This is extremely sensitive area, and yeah, it's a fascinating process. So I covered that. Let's see, how long have I been talking? Doesn't show. No. Okay. So with that being said, let me talk about temporal and spatial summation. And this is another area which is interesting. Let me change the page. Draw like rectangle. Okay. Okay. And then we have that. Okay. And let me, I want to explain another phenomenon, which is summation. Summation. So in summation, we have temporal summation, and then we also have spatial summation. And these two have a different processes. So now let me go ahead and draw a different axon terminals right here, right? And then I draw a bigger. I'm gonna draw a dendrite like this. Dendrite of another cell, maybe different color. Dendrites of another cell like this. Okay, now we know, based on what I mentioned previously, that these neurons or these axons, dendrites and axon terminals, they talk to each other. They send information through the synaptic cleft, and so once what happened is that when the action potential goes and when the signal is sent to this cell, if I wanted to graph this, so we know that we have a negative 70 millivolts difference, or that's our resting point. And then let's say if in order for action potential to happen, is it happens at the action potential happens at negative 55. We call this a threshold. The threshold means the minimum requirement for a cell to feel some discharge in order to have that action potential. Anything below this, the cell doesn't respond, doesn't provide that action potential. So now when the information is sent, when a signal is sent to the other neurons, we see on a graph that there's a little bump right there, right? But when the information is going after each other, right after one and after each other, so it causes this little displacement. It adds up, and I am not doing a good job of that. Let me see. So it adds up. So these little action potential adds up, the discharge, it adds up until it reaches the threshold of our cell. And once it does, it goes to the full action potential, and now it comes back, and then hyper depolarization, polarization, and back to the resting area. And that's, so this is the depolarization, this is the repolarization, hyperpolarization, and back to the resting area. And this is really important. This phenomena, when one neuron transfers the signals to another neuron, right? This is called temporal. Because why do we call it temporal? Because it transfers from one neuron, and it's a matter of time that these little discharge displacement gets built up or adds together in order to give us that action potential, right? So that's temporal summation. Now let's talk about spatial summation. So again, we have, I'm going to use the same color. So I'm going to use this. So we have, in neurons, axon terminals transfers the information to, another dendrite or another cell. But here we have another neurons. I'm just going to draw one. Maybe this it can be more. It's not limited to one, but just for the simplicity. So we have another neurons that these neurons, these axon terminals, also send information to this neuron, right? Also send information to this neuron. So now this dendrite or this cell receives the information by two axon terminals, and this is what we call, a spatial summation. And because the neurons receive, by separated space, it receives two neurons firing, two axon terminals firing. It receives signals from two different or more neurons, right? So that's summation and temporal summation. And then the last thing that I wanted to cover is agonist and antagonist. And this is exactly how the drug works in our brain. Some drugs are agonist. So let's say if we have, let me open up a new page again. I want it in black. Let's do that. Okay. Let's do something. Okay, now we have agonist and antagonist. I'm gonna do agon. Did I pronounce it? Yeah, agonist. So we have agonist. An agonist, what agonist is, for example, we have this membrane, and then we have, let's say we have our channel, ion channel, that let's say this is a sodium channel, and so it has a little receptor. I'm drawing it completely different than we did before, just for better understanding. So agonist is if we have we have a dopamine right here. This is a neurotransmitter, dopamine, floating around. Something that is look like a dopamine, right? Something that is look like a dopamine will come and attach to these receptors, right? It is not a dopamine, it's an agonist. It's something similar like the drug. I don't know, what is the drug? Wellbutrin. That it comes and attached to these receptors, and so it causes the sodium ion to pump in a sodium. It opens the, so once this agonist attached or to this membrane, the ion channels opens up and causes this sodium to float in, right? Na. And it mimics the behavior of the actual dopamine, or in this case, or serotonin or norepinephrine. And we have another thing, antagonist, that so the antagonist is does the completely different thing that agonist does. So let's say again, we have this channel, we have this receptors. So if agonist mimics the behavior of neurotransmitters, then antagonist, it doesn't do that. It does the completely it has a completely different effect. So what if we have a so this is where most depressive drugs come into the play. So if we have, let's say, a serotonin neurotransmitters here, right? So what antagonist does, it goes and it inhibits the reuptake of that serotonin or that neurotransmitters. So it keeps the gate of this ion channel closed by attaching itself to these receptors and letting serotonin to be abundant in a synaptic cleft, right? So that's the role of agonist and antagonist. Yeah, and that will covers it. So if you have, I know it's a lot to take in. A good resource is going to be Dr. Berg's lectures and also the book, and also Inquisitive. If you have any questions, if you have any concern, if you didn't understand anything, please do send us an email. Please go ahead and start reading the chapters. There's a lot more material. Questions could be more difficult, so don't wait till the last moment, because with the remote teaching things, it's easy to get behind. So yeah, and if you had any questions, any concern, again, you can always send me an email, or if you wanted to set up a private office hours, I'll be available. And with that being said, yeah, have a good one. Take care. Stay safe.